All right, great to be back here. Third session of the day. We have a very, very diverse panel today, but in all cases, this is a mix between understanding evolutionary processes and understanding what's going on in this world, and how can we engineer and understand the world better through this. So the first speaker of the day, David Bikard. He is the first iGEMer in France, iGEM 2007, best prize, what was it, foundational track prize for the first participation of a French team. Thanks so much for being with us. David Bikard from Pasteur Institute.
So yeah, it's really amazing to be back after so many years and see iGEM still going on so strong. And also great pleasure to, how do I pass my slides? I think I need this, right? Yeah. Thanks. Amazing pleasure to tell you about some of the work that we've been doing in my academic lab at the Pasteur Institute, and also some of the work we've been doing with Ezhil Bioscience, a biotech I co-founded. And so over the last years, we've been part of the community developing a lot of the amazing CRISPR-Cas tools that people now use for a lot of different tasks. And in bacteria, there are, let's say, three main things people do with those CRISPR-Cas tools. The first one that was actually developed was when we realized that if you program Cas9 to cut in the chromosome of bacteria, you will kill these bacteria very efficiently because they don't know how to repair the type of breaks that this CRISPR makes. You can, of course, try to control repair and obtain different edits. You can do genetic modification also with new generations of CRISPR tools, and in particular, base editors have been extremely powerful and well adopted in the field. So what a base editor is you simply fuse a deaminase domain to the Cas protein, and what that will do is that it will chemically modify a base, like a cytidine, turning it into uridine. And so you can make very targeted base modification. And you do that with Cas proteins that you inactivate so that they don't cut the target DNA. And then you can, of course, control gene expression by either silencing genes or activating promoters. So there is a lot of things we've been doing around that field and developing those tools, but also using them, and in particular, using them for high throughput screens. And this is leading us to a lot of novel insight in bacterial genetics. And so you might already be familiar with the concepts behind those CRISPR screens, but the idea is that you can design libraries of guide RNAs that are going to target all the genes in your bacteria. You order them, obtain on-chip oligonucleotide synthesis, so you get a tube where you all these guide RNAs, you clone them on a plasmid, and you put that into your organism, so in our case, just E. coli. And then you induce the expression of Cas9. It's going to silence all the genes in the genome one by one, and you just screen by growing your bacteria in the condition of interest. And then you look at whether guide RNA are enriched or depleted. If they're depleted, it means that probably they blocked the expression of an essential gene. If they're enriched, it means that they gave somehow a fitness advantage to the bacteria. And so this is a very nice and high-throughput way to do functional genomics in bacteria. And what we've been doing in my team recently is trying to apply these tools to start and study the genetics of bacteria in the microbiome, which is such an important ecosystem for health, and also such a complex ecosystem with so many things we don't understand and that we need to understand. So my PhD student, Amandine Mer, has been constructing these guide RNA libraries for different strains of E. coli, and then introducing them in the gut of mice, and trying to see what kind of insights we can obtain from these screens. And so she's been doing experiments looking at different diets, trying to understand what are the genetic requirements for bacteria to grow depending on the diets given to the mice, looking at different strains of E. coli, either commensal, pathogenic, and also looking at inflammatory conditions. So I'm not going to give you all those results. I just want to give you a very quick snapshot of what the results look like. So here you have a genome browser view of some of those results in this diet experiment. And each point here is a different guide RNA given to a mice in a different condition. And so, for instance, here it illustrates a very interesting result, which is that when you block the expression of a fructose utilization locus in E. coli, the bacteria is not happy, but only when you're doing that in the high fiber diet. And so what's quite interesting here is that this is done in a controlled diet, and we know that there is no fructose given to in the diet of the animals here. So what this tells us is that the E. coli must be somehow utilizing fructose produced by other members of the bacterial community, and so this is pointing us to some very interesting cross-feeding interaction that are happening in the microbiome. So this just tells you how this type of screen is very rich in information about the genetics of the bacteria, but also what the environment of the bacteria looks like. This is just another quick snapshot showing you results on a screen we performed in inflammatory condition. And if you look at the PCA plot on the left, what you can see that all the red points correspond to the CRISPR screening results for mice that were given that were either under inflammatory condition in red or in a healthy state in blue. And you can see that those separate very well. So what that means is that just looking at the results of the CRISPR screen, I could tell you was the gut in an inflamed state or not. So you can really think of this also as a kind of a very powerful diagnostic strategy where you could think about your E. coli as a sensor of its environment, and that's reporting through these CRISPR screen results. So something now we are very keen on doing is being able to deliver all those amazing CRISPR tools to the bacteria, not just in the laboratory setting, but directly to the bacteria in the animal gut. If you can do that, you can go and study the genes of the bacteria directly in the natural environment, and you can also maybe start to think about possible therapeutic intervention targeting bacteria in the microbiome. And this is work that we've been doing with Eligo Bioscience. And the strategy we're using to deliver this CRISPR-Cas tool is by vectorizing bacteriophage vectors. So we take the capsid of the bacteriophage and we package inside a phagemid. So it's just a plasmid on which we can put any gene we want. And we're interested in putting different kind of CRISPR tools, including CRISPR nuclease to kill target bacteria that might carry variant factors, antibiotic resistance gene, and specifically eliminate only the bacteria in a sequence-specific manner. We can introduce base editors to make specific modification without killing the bacteria, or we can think also about delivering therapeutic genes. But I want to show you some of the results we published recently on base editor and demonstrating how it's actually possible to genetically modify a whole population of target bacteria directly in the animal gut. And we've been for this engineering a phage that you might know. It's the phage lambda, so probably one of the best studied bacteriophage. However, we realized that if you just take phage lambda and try to deliver DNA to E. coli in the gut of mice, it just doesn't work well at all. And we figured out that the main reason for this is that lambda uses this lambi outer membrane porine as a primary receptor. However, E. coli in the gut environment tends to not express lambi or even evolves to break lambi. And so we had to engineer phage lambda, engineering the primary receptors to change it from lambi to OMC, engineering also the cytal fiber so that they recognize the LPS of E. coli. And doing all those modifications, we were now able to deliver DNA very efficiently to E. coli in the gut of the animal. And then we've been delivering base editors, so either adenine base editor or cytosine base editor, so that we can make different types of modifications. So just as a quick snapshot, those are results we obtain with this strategy, trying to inactivate an antibiotic resistance gene in the chromosome of E. coli. And so we first colonize the animal with this antibiotic resistance E. coli, then we treat with this oligobiotic, so this phage mid with a base editor that we programmed to inactivate the resistance gene. And you see that already with a single very high dose of the treatment, we're able to modify close to 100% of the bacteria directly in the gut environment. And what's very interesting is that if you give multiple treatments, so on the bottom left, you see we give like 3 times an intermediate dose, and we increase editing efficiency over time. But when we stop treatment, the bacteria in the gut, they stay edited. And that's because the edit we're making in this context, it doesn't give any fitness defect to the bacteria, and it's extremely stable over time. So we can have this permanent modification established in the gut microbiota. So final story I'd like to tell you is a recent novel interest of my lab, where we've been working on a fascinating genetic element called diversity-generating retroelements. So just out of curiosity, how many people in the room have already heard about these DGR elements? So yeah, maybe only a handful of people. And so my hope is that this might be a kind of new CRISPR, so not there yet, but it's really a fascinating genetic system that's naturally found in some bacteriophages, where they've been discovered by a guy called Jeff Miller at UCLA in the early 2000s. And what these elements do is that they introduce targeted hypermutation in a variable region of the phage receptor binding domain. And what that lets the phage do is that it accelerates the evolution of the phage so that it can very quickly adapt and evolve to bind new receptors and infect new host bacteria. And the way this works is actually through a very interesting retroelement, where you have a small non-coding RNA called the DGR RNA that carries an imperfect repeat of the variable region. And this non-coding RNA is reverse transcribed by a narrow-prone reverse transcriptase that makes specifically errors on adenine residues. And like this, it's producing this mutagenic cDNA that can then do through a recombination, replace this variable region and introduce a stunning amount of genetic diversity. And so it's been estimated that among the known biological mechanisms for genetic diversification, DGRs are capable of exploring the highest ceiling of coding sequence variability. So a breakthrough in my lab recently is that we've now been able to actually take a DGR system, make it work in E. coli, and reprogram it so that we can now go and diversify any target sequence we want. And so this is programmable, targeted, in vivo hypermutation, which I think can find a lot of very exciting application in directed evolution setups. And one of the things we'd like to apply it for is connecting back to a previous part of my talk, where we want to deliver DNA to bacteria of the microbiome, is now we can take this tool and use it to evolve phages that don't naturally have DGR systems and try to now adapt them to deliver DNA to bacteria of interest. And so here's just an example again on bacteriophage lambda. So we're a bit here recapitulating the arms race between the receptor and the receptor binding protein. And so we first turn the DGR to LAMB, which is the receptor of lambda, and we select variants of LAMB that lambda cannot recognize anymore. But then we turn the DGR to lambda itself, and now we can restore the ability of lambda to infect the bacteria that carries those mutated receptors. And if you look at the mutations we obtained, they map nicely at the non-interface between the proteins. So this is everything I wanted to show you today. I'd like to thank the member of the synthetic biology group at the Pasteur Institute, and also a big thank to the team at Eligo Bioscience, who did all the work on the in vivo base editing. And thank you all for your attention.
Our next speaker, Aude Bernheim, also from the Pasteur Institute, also an iGEMer, world champion 2013. Aude Bernheim, defense mechanisms in bacteria against phages. What can we do with them and how do you find them? Thank you very much for being here.
Thank you. So, hi everyone, my name is Aude Bernheim. My lab is at the Pasteur Institute in Paris. And I'm very excited to be here, surrounded by iGEMers, because as Ariel said, I participated in iGEM, and that was very important to me. And several of my teammates from that year are in the audience, so it's super cool to be able to hear that with them. And so today I will discuss the evolution of immunity across domains of life. So I'd like to start with this sentence from Jacques Monod, a French scientist, who said what is true for E. coli is true for the elephant. And in that sentence, Jacques Monod captures the essence that some of the processes that are existing in living organisms are shared, and that by studying bacteria, we can somehow study some of these very fundamental processes. One, I would say, domain of biology where this did not seem to apply is host-pathogens interactions. Because yes, all cells, bacteria, eukaryotics, such as plants and humans, face pathogens. However, there is this idea that there is an arms race between pathogens and cells that leads to the diversification of immune mechanisms. And this is a picture that we've been working with for a long time. The idea is that bacteria would have defense systems, such as CRISPR-Cas systems, or restriction modifications that are very different from the type of systems that are present in plants or in humans. And interestingly enough, the study of bacterial immune systems led to two biotechnological revolutions that are restriction modification and CRISPR-Cas. But this paradigm that bacteria had two major anti-phage systems and that bacteria do not share any immune mechanism with eukaryotes has really started to change thanks to recent discovery in the field of anti-phage systems. And it all started with studies first initiated by Eugene Koonin and Kira Makarova in this wonderful paper that noted a very peculiar observation. And the idea is that they looked at bacterial genomes, and directly in these genomes, I cannot point, but they saw that actually you had systems that cluster together near one another. And they call this defense island. And when you look next to these known systems, which are colored here on the scheme, you actually had genes which are beige and of unknown function. And the team of Rotem Sorek developed, in my favorite paper, a method to actually use this very peculiar phenomenon in bacterial genomes to discover new defense systems. In this wonderful paper, it's my favorite paper ever, so you should check it out. They use it at scale, and in one shot, discovered more than 10 novel, completely new anti-phage systems. This spearheaded a field that I'm now part of, which pretty much set for a completely different understanding of bacterial immunity, and as you will see, of eukaryotic immunity too. Why? Because since that paper in 2018, over 250 novel anti-phage systems have been discovered, with dozens of novel molecular mechanisms that are more crazy one than another. And so really there is this great question of how much diversity is there to uncover. But maybe the biggest surprise that emerged from this was that among all these new anti-phage systems that were discovered, some bared a striking similarities to known eukaryotic, including human immune mechanisms. You can see overlapping structures of the human viperin, a gene known to be involved in immunity in humans, and of a bacterial viperin that we discovered. And they're the same molecular machine. They function the same way. And so somehow it seems that they are part of immunity that have been conserved from bacteria to humans. And so there is a paradox from an evolutionary perspective between this arms race and adaptation to pathogens and these really ancient building blocks. And so this is what my lab is studying. And, oh, sorry, I forgot to mention that the idea is that with the viperine and other examples, it first emerged that they could be exceptions, but then, you know, one exception plus one exception plus one other exception is not an exception anymore. So we decided to name these ancestral immunity for the immune modules that are shared by prokaryotic or bacteria and eukaryotic immunity. We know of more than 10 examples, and it's just the beginning. And so this is what my lab is studying, is trying to understand the fundamental principles that govern immunity across the domain of life. And on one side, we are really interested into uncovering all the diversity of bacterial immunity with diverse questions, and I'll give you a hint of the type of things we can do in my lab around this. And on the other side, around, I would say, ancestral immunity. So the first thing that I think was a big question is how many of these new anti-phage systems bacteria encode. So we developed a software to actually do this, and we've been updated Exist because every year there are more and more systems being discovered. And so right now we know that a classic bacteria will encode more than 8 immune mechanisms on average. But there is this one bacteria that encodes 65 different immune mechanisms, which I think is a wonderful diversity to dive in. And why is this important? Well, for example, you can look at these defense systems and use what we know from genomic information to go and train algorithms, both using genomic data and phage-bacteria interaction that you can actually compute, to then recommend, for example, tailored phage cocktails. So this is something we were able to do in a recent paper and show that we are able to recommend phage cocktails with more than 90% accuracy on bacteria that the algorithm had never seen. So this is what you can do by starting to understand the diversity, but maybe the most exciting thing has been a bit hint by David before me, is what you can take maybe that could be then, you know, applications from bacterial diversity. And so here I'll just give you one example of one thing we're interested in my lab. And the idea is that it all started by looking at natural products, which are small molecules produced by bacteria and that have been harnessed by humans to pretty much do antibiotics or anti-cancer and stuff like this. And we wondered whether in this gold mine that these are these natural products, whether we might be able to find antiviral molecules. Why? Because of this discovery of viperine and the fact that immunity is conserved, you can imagine that some of the molecules that are used by bacteria to defend themselves against their viruses, some could actually be used to defend human against their viruses, and this is the case of this molecule called DDH nucleotide. So in the lab, we set up a pipeline to discover this at scale through a systematic exploration of the capacity of bacteria to synthesize antiviral natural products. The pipeline just overlaps bioinformatics prediction of what we think will produce a small molecule and what we think will be antiviral. We then take candidates, have anti-phage bioassays, and identify the molecules. And so to mine diversity, I just want to mention that we can always think of different ways. So with Vincent Libis within the room and with Ariel right there, we devised a citizen science project to go and collect soil and mine diversity. And it was really successful because people all over France actually sent us soil to go and mine these soil bacteria from which we find the most diversity of potential antiviral molecules. And that was where to mine diversity, and the second step was to get a proof of concept. In this first paper that you can find as a preprint, we were able to discover a huge family of lentipeptides that have an anti-phage function with a very cool mechanism that pretty much you have this peptide that is modified and is able to specifically target the phage cycles by preventing the phage from starting DNA replication through what we think is some form of a molecular glue mechanism by gluing it to another proteins. And so this is really an antiviral molecules produced by bacteria. We are now scaling up this process, and we have identified hundreds of families of genes that could produce this type of small molecules, not lentipeptides, but many different chemistry, ranging from nucleotides to indeed peptides or other things. So right now we are investigating and mining this. And Finally, on the diversity side, I wanted to say that a big question that is in the field is how much diversity are we talking about? I told you that we went from 2 with CRISPR and restriction modification to then 10 with this first paper, today 250. Well, recently in my lab, we adapted algorithms that are used by all these natural language processing type of algorithms, so-called LLM, to actually take either as input protein sequences or the genomic context of genes to predict or not whether they would be involved in anti-phage defense. And using this, we were able to predict that more than 45,000 families of anti-phage systems are out there to be discovered, and it's kind of a wonderful goldmine and roadmap to go and hunt, and we're making these predictions accessible to the community, because of course at the scale of my lab, we won't be able to go and pursue all these 45,000 families of anti-phage systems. So this was on the diversity of bacterial immunity, and now I kind of want to switch gear and tell you how we think we can now use the conservation of immunity across domains of life to discover new important immune functions. And the key idea here is that these ancestral immune modules that I introduced to you in that presentation, which are the domains or the proteins that are shared between bacterial and eukaryotic immunity, were all discovered by taking things that were known in human immunity because it was well studied, and then we discovered new things in bacteria and mapped the knowledge from humans to bacteria. Well, in the lab, with all the amazing developments that have been happening in bacterial immunity, we thought maybe we can do the reverse. We can start from what we know about bacterial immunity and discover new things about human immunity. And I'm doing this in a great collaboration with the lab of Enzo Poirier at Institut here in Paris. And we have a couple papers that have been published, but I just want to highlight the most recent one, and it's a story that was co-led by Delphine Bonhomme in Enzo's lab and by Hugo Weizmann in my lab. And it goes like this. I'll just give you the gist of it. You have a protein domain called SIR2. This protein domain is involved in a family of proteins called sirtuins that are not involved in immunity. And in bacteria, more recently, it was also involved, this domain, in anti-phage activity. So what we did in the lab is to detect all the proteins in genomes, bacterial, archaeal, eukaryotic, and try to see if something were missed. And what we observe in this phylogenetic tree is that the ring around the tree, if you see that it's green, it's a eukaryotic homologue, and in blue, a prokaryotic homologue. And the tree really splits in two, where in both big subclades, you have bacterial and eukaryotic homologues. And when you map what is actually known, you see that all the sirtuins are in this light pink clade. So all the non-immune proteins are in that clade. But when we map the known and anti-phage, so immune proteins in bacteria, they are all in this dark red subclade. And so somehow it seems that phylogenetically, all the immune proteins are there. But among all the blue proteins, you actually have some green, so some eukaryotic homologues, and notably a homologue that is a human homologue called FAM118B. This gene was uncharacterized, never linked to immunity. Our hypothesis, based on this phylogenetic tree, was that it might have an immune function in human. We tested this hypothesis by doing a very simple experiment. The idea was to challenge murine macrophage with LPS and monitor inflammation. The way it goes is that if you actually get LPS stimulation, you have a dose-dependent response with nitric oxide production, as shown here. When we did a knockout of these for FAM118B, that I will now call Ciral for Cir antiphage-like, this is what we got. No more responses. So we were very much surprised by such a strong result. So our first thought was, of course, it's an artifact. But then a lot of work from Delphine and many people in Enzo's lab were able to show with many bar graphs that I'll spare you that actually this holds true and that actually Ciral is important for the TLR pathways in general, so several TLRs, and that it's important both in a mice setup and a human setup. And so this pretty much constitutes a new target for immunotherapy that we're now investigating. But what I got also very excited about, and if you like me, when you think of eukaryotes, you actually think that it's a diverged clade of bacteria, because that's what they are. Pretty much you can imagine that you have one data point in this clade of eukaryotes. But what about others? When we did a more detailed analysis of all the SIR-IM proteins, so the proteins that have this SIR2 immune domain, you can see that actually there are many, many, many living forms that have this, including koalas, but not kangaroos. Why, I don't know, under investigation. Fish, or actually small unicellular eukaryotes that are very important in the oceans. And so somehow, by looking at these conservations, we can just project what we know, not only for health, but also for questions linking through ecology. And so this was very exciting. And of course, we are now investigating different things on SIR-IM, like the fact that it's a novel actor of mammalian TLR pathway and that we can harness it for therapy, but also how we can use our knowledge to just understand what's going on in immunity in many eukaryotic organisms that has not been studied for immunity yet. And so, of course, the big question is how many ancestral immune domains are there? Well, I cannot give you a precise number yet, but I can tell you that we have been identifying dozens of ancestral immune gene families. Right now in the lab, we know that there are dozens of them, and we have used them to actually predict eukaryotic homologs, thousands of them, across different organisms, and are collaborating with labs doing plant science, algae science, of course, human immunology to test these predictions. And so we're very excited to see where this is leading us. So I just want to take one slide to conclude on how do we reconcile these somehow two very different evolutionary dynamics. One of diversity that I showed you and that we want to mine, and one of conservation. And so if I started with François Jacob, for those who are following the French scientist, with Jacques Monod, I'll end with François Jacob. So François Jacob put this idea of molecular tinkering, that pretty much you have building blocks, and you can think of biobricks here in this area, but you can have building blocks to build new things. And somehow, the way that I think about these ancestral immune modules is that you have building blocks to build molecular machines dedicated to immunity across domains of life. And some of these building blocks are actually conserved throughout domains of life. And so if we are able to discover them in bacteria, delineate them, and then be able to follow their evolutionary history, it's very a powerful method to actually predict immunity and understand it at different scales. And so overall, I feel that, I hope I convinced you, that this makes that ancestral immune modules are a reservoir to build immune systems, and that bacteria are a great model to study immunity. And on this, I want to thank my great lab, because we work in a very collaborative manner, so everyone contributed to the studies I showed, my great collaborators, and you for listening.
Thank you so much, Aude. Next speaker on stage is Ron Milo, professor at the Weizmann Institute. Ron can address any question that he can put a number on. Okay? So if anyone knows by your numbers, by Rob Phillips and Ron Milo, that's the first thing you need to know in order to learn biology. And if you want to solve problems, you need to frame them correctly. And so from a scale from sort of carbon fixation to the surface of Earth, right? Ron, floor is yours.
Thank you very much, Ariel. I have to say I had a lot of expectations coming here to the iGEM. I heard so much about it, but coming here, it's beyond what I imagined. And for that, and you know, every time there's a break, you know, the energy around here is so amazing. I'm getting new ideas. So for that, I really thank the organizer for thinking about combining the SB8.0 with the iGEM. So Ariel, Matthew, Drew, I think you all deserve like a standing ovation from everybody here. And I'll be happy to share with you, you know, the vision of what we're doing in the lab and how we're trying to quantify our way towards that. Maybe I should start with some sort of combination between apology and warning that I think maybe holds for other things. What I'll show you, I have, you know, less than 15 minutes to show you what we worked over for more than 15 years. So I think very often I find that for me, when I hear such talks, I get the imposter syndrome, feeling like everybody else, you know, understands what we're talking about, but only me is not following everything. So if you feel the same, you're not alone. It's very common. And with that, I'll move to the essence, and I'd like to start with sort of like a motivation in what drives our work forward, and that's thinking about the balance between humanity and nature and the biosphere. And it's nice to take a concrete example. Let's think about mammals. You know, we are part of that family, and we're also inspired by mammals a lot. So if we were asking what fraction of the mammals that are currently on Earth in terms of mass is wild, both in the overland and in the ocean, all those we see on all the BBC documentaries, etc. So on that, we worked pretty hard, and what Lior showed in her work is that currently only 5% of the weight of mammals in the world is wild, where all the rest is us and our domesticated livestock. And if you ask yourself, how did we come to this situation in 2025? So just this Monday, we published this paper that shows the evolution from 1850 until today, how the weight of mammals in the wild has been decreasing, and the weight of us and livestock has been increasing rapidly. And I'm happy to see people taking photos, and anybody who wants, I'll be happy to share the whole presentation. It's all in order to pass the message to as many people as possible. And this is not only just like a graphic that's interesting from a intellectual perspective. It has many implications because these livestock are also at the heart of many of our environmental problems. It's a major driver not only of climate change and greenhouse gas emissions, but also in the land use change. So the largest fraction of the land that we're using on Earth is associated with those livestock. It's also what we're using much of our water resources, the eutrophication that leads to dead zones in our water and antibiotic use, etc., etc. So in a way, what you see here is what's feeding into a large fraction out of all the projects, problems, solutions that people are thinking about in something like the iGEM. And so with that, let me share with you, one effort that we've been doing, and that is like trying to think about the future of what could be alternative both to these livestock and in thinking about our challenges for supplying our food in the future. And that is thinking about how microbial production could be used for food production and as well as other uses. And that could have wonderful not only environmental benefits, but could also be very advantageous in terms of nutrition that might be good for us more than what came out of evolution that is not necessarily the best for the 21st century and how we like to eat our food, as well as to being resilient to all sorts of challenges that it seems like we'll be facing and would require resilience a lot in the future. So just a short recap. When you're thinking about our biosphere, you can break it down in your textbook into two parts. Those that are the producers, the autotrophs that take carbon dioxide and energy from the sun. They give us all those nice organic molecules that then serves the consumers just like us, the heterotrophs that basically build upon their hard work. And in a way, this is really the most fundamental process, and even though there's all sorts of sponsors associated with this meeting, really the sponsor to this meeting as well as to your career and to the whole civilization of Earth is really this process of doing carbon fixation, part of photosynthesis, taking carbon dioxide, and with that coming up with like a really ingenious way in order to store carbon and store energy. And there are two approaches in trying to think how can you find an alternative for this or to think about microbial production. One is to leverage natural autotrophs, such as cyanobacteria, algae, etc., plants. And the other is to try to introduce carbon fixation into model organisms. So both things are of interest, and I'd like to focus on the second approach. And that is to try and ask whether we could take E. coli, whose diet is based on eating sugar and releasing CO2, not unlike how we run our life and our diet, and to ask whether on a laboratory timescale with some rational design we could change the diet of this model bacterium to eat carbon dioxide and produce sugar. When I'm saying produce sugar, it's really from sugar. Biochemistry could achieve anything else you want. So basically take carbon dioxide and leverage that to produce whatever you're interested in that biology knows how to produce. So this was the question we set out to investigate. And just to be clear, we're not talking about, redoing all of photosynthesis. We're focusing just on this part of the dark reactions, although talking with some people here, we could now, I think, maybe be even more bold about this. And in order to supply the energy, we're doing that using fomate, which is a substrate that E. coli could not live off of, but could extract the energy from. So this is also, this ability to do this is also known as the Calvin cycle, and it has like 13 components, very, very challenging, but it could have been worse. It could have been much worse, because actually almost all of the components are already within the metabolism of E. coli. Only 2 genes are missing. Rubisco, the key enzyme that's the most abundant enzyme on Earth, and the friend of it's PRK that produces the substrate. So in principle, you could take those 2 things, put it on a plasmid, transform it, and basically you could hope that now E. coli could do carbon fixation. This was, stage 1 point whatever, and this is how we started. We're supplying it with fomate, and the short answer is that it failed. And we already got the grant from the ERC. The PhD students were deep at work. We had to find a solution, and for that we decided to try and combine the rational design that we did together with the most powerful tool that our dispense, and that is evolution. So we put it into a chemostat, which is really the machine where if you want to put really strong selective pressure and supplying it with time, you can hope to maybe change its diet. And therefore, we did this challenge to E. coli. We said, you know what, we'll put you in the chemostat where you're really starved, like, super starved. And at the same time, we put you in a heaven of lots of carbon dioxide, lots of energy, so we're supplying you with everything you in principle need, if you only be willing to adapt to becoming autotroph. So this was the idea. And long story short, very long story short, because usually each such experiment takes about half a year and ends with contamination, so then you have to repeat it again, and then it takes another half a year, usually another contamination, and then another half a year, and then you check and you see some colonies. And what you expect now, and that's what the reviewers expect as well, is those colonies are not what you wanted, but the E. coli had so long time, such a strong selective pressure, it found some way to eat something from the plastic, from the water, from this, from that. So even though we think that now we have growth or something like that, we have to convince ourselves. So how do we do that? The way to do that is we really want to label carbon dioxide to see whether it's coming really from the air, whether it's really autotrophic. Now, it's not so easy. If you put a GFP on carbon dioxide, it doesn't work, right? CO2, you don't have a lot of space there. So how do you still label it? You can use a stable isotope, so that's pretty neat. You can just label it with another neutron. And with that, you can really follow. So what you put, like in the airspace, you put carbon that has another neutron, and you just let the cells grow, and you're looking within the metabolism to all the different components, and you try to see if they're purple, meaning they came from, something natural like the fomate, or maybe they took it from, any other contaminant, impurity, antibiotic, which we made sure not to have, or anything else. Or if it would be green, it would be coming from the air that you put, which is labeled carbon dioxide. And the short version is that if you look at this, everything is green, except for the fomate that we're putting in. 100%, which made Shmuel Gleiser and Nir Antonovsky pretty happy, because Niv came to the lab saying, you guys, you have these crazy ideas. I'm willing to spend my 1 year of masters to show that you're wrong. So he spent 1 year on that, couldn't show that we're wrong, couldn't show that we're right. We spent like 7 years on this. At the end of his PhD, was able to show that and passed the baton to Shmuel, which continued working on that, because he made only the Kelvin cycle, not the whole thing. And with that, they became pretty happy. And we were pretty curious now to understand what happened in the half a year to a year of evolution that was happening in the chemostat. So, of course, we sequenced everything, and could find all sorts of different mutations, several dozens of them. From that, none of them is the targets we expected, not in Rubisco, not in the Kelvin cycle. Really rational, nothing of the rational expectation worked. So we spent several years trying in all sorts of nice ways to understand what happened there. The short version is we understand it could be done with only 3 mutations. Out of the dozens and dozens of mutations, in a repetitive manner, we could shrink it down and show with the work then of Ro'i that continued from Shmuel and Elia that continued from Ro'i, that only 3 mutations are enough. And with that, we could recapitulate autotrophy and really show that like this textbook situation of, autotrophs and heterotrophs being in a completely different part of the textbook, you can transform from this to that with just a few genetic steps. So if you're interested, this is a summary of some of those findings. And I think the question now is, so we understand there's like this, vision one could think about valorization, taking carbon dioxide, using it for all sorts of cool things. So basically the idea would be to take direct air capture or something flue gas from our industries, be it, iron smelting or making cement for buildings, together with renewable energy that happily costs less and less, combine that with microbes in order to give us the things that we need, be it high-cost foods and chemicals, staple foods, or fuels. The question is now, is that economically viable? And for that, we wanted to combine with that with some understanding, like a techno-economic analysis of this project. So let me touch on that very quickly. So is that something that could be economical? So the short answer, also on that, you can do a back-of-the-envelope calculation to give you some insight in a transparent manner on what is happening here. And so you could look at what would be the cost of your energy supply, how much are you willing to get a credit for the carbon. And what happens is that even though we want to valorize carbon dioxide, we know it causes climate change and many other issues, the amount of money that we're willing to pay for that, that's about, if you're really willing to pay a lot, it's on the order of $100 per ton. That's a few cents per kilo, which is very little in comparison to the cost that would require you for the energy demand, which is on the order of $1 per kilo, and the cost, the capital cost that's usually associated with even the most mature technologies like, making beer or bread or any other big fermentation. And therefore, if it costs you about $2 and you get just a few cents for the carbon dioxide, it means that the product that you need to then sell would cost at least a few dollars. And now you ask, how does that compare versus the things that we're interested in producing because humanity needs them so much? So the short version in the analysis that recently came out from Sam Lovett that came to do this work in my lab is that if you look at high-cost foods and chemicals, that could be very promising. And therefore, there's a big market. And it could also help, if you remember, the issue of livestock. That's also the price that we're willing to pay, for example, for beef. So there's a good potential there. In terms of staple foods, it's borderline because the amount of price that we're willing to pay is usually on the order of that or less. But maybe humanity would understand that given the externalities, all the costs associated with those environmental burdens and some subsidies that we're putting, maybe they should be shifted in order to make the economy right. And I think this is something that would necessarily have to happen at some point. And if you think about fuels, we're very far off. So I think this takes us to a more quantitative grounding of what is realistic and what is less realistic and what is probably not realistic. And I think we should be pretty transparent about that. And if you get to a different conclusion, I'll be delighted to talk about it and understand. And in closing, I'll just like to wrap it up and say, on the one hand, I think transforming diets is really one of the greatest challenges of humanity. And if you like it or not, maybe you'll prefer to enjoy the croissant or the wonderful dinner that you're planning for tonight. That's all fine and well. But in terms of 8 billion people, in the longer term, it's unclear, if not impossible, how to continue in that way. And therefore, we have to find alternative ways. And I think maybe better ways in terms of nutrition, in terms of resilience, etc. And definitely lighter on our nature. And our work, I think, is a combination as a community, is to try and understand that better and to try to move in that direction as fast as possible. And that is only achieved by people that are willing to go on this bold voyage of starting, as you can see here, with the ideas of the late Aaron Bar-Even and Elad Nur that came up with those ideas and tried to push on that together with a wonderful team that I mentioned some of those people. And you start with that and the ERC believes in the idea or even pushes you there because you know you need to offer something that's far-reaching. And with that and some more people that are willing, for example, Shira, this is on her wedding day, she was willing to start the evolution not in a chemostat over half a year, but to do it manually, every day to put the bacteria starting that way. And at this point, she was pretty happy, both because it was her wedding day, but also because we moved it into the chemostat, so she didn't need to do it manually anymore. But this was, as I told you, it wasn't the end of the story. You had another round of bold students that had to push it even further. And then finally, those people are happy because now we're pushing it now to trying to use carboxysomes in order to work also in ambient air and to try to move it to other bacteria like Bacillus subtilis, which is what the SNIM there is doing, and trying to push the limits from there. But we really need, they say you need a village, so you need a few villages like we have here in order to make that work in the longer term. Thank you very much.
Amazing. Thanks so much, Ron, and we'll stay with this vision with Ezhil Subbian from the CEO of String, of what can we do with carbon dioxide and why is it so important? Thanks so much.
Hi everyone. Great to be here today. Fantastic energy in the room. It's my first time at iGEM as well, even though I've been part of the synthetic biology ecosystem for quite some time. And so thanks for having me here today. So I'm going to take a slightly different, not even slightly, I think a very, very different tone to what we've been hearing so far. And given it's the end of a very long day, I'm not going to talk as much science. Apologies for that. What I will talk about is the work that we've been doing in terms of redefining manufacturing. But I also perhaps want to set some questions forward for this audience here, kind of perhaps sharing with a community of like-minded people to see how we could take more and more ideas and create bigger impact. So before I begin, just a little bit of background about who I am, what we do, so you can also perhaps have some context to where these questions and where these learnings are coming from. So I was part of the synthetic biology ecosystem in the Bay Area for a decade plus. Worked with companies like Codexis and Gevo, where we built very, very interesting tools to convert sugar-based feedstocks into molecules for the ethanol and biofuel processes. And then around 10 years back, decided to move to Bangalore, India. And for the last 10 years, been building a startup called String, based out of Bangalore. At String, what we do is actually, we've kind of addressed the question that Ron brought up to say, hey, can microbial production be economically viable to make food? Because that's essentially what we do, is we've taken greenhouse gases, such as methane and carbon dioxide, and via a fermentation process, a bacterial fermentation process, we convert this into proteins, into peptides, into small molecules. And we've now commercialized these solutions for different end-use markets. For example, we now have a very, very interesting protein powder that we make using methane via a bacterial fermentation process. We have peptides that we've commercialized for the agri sector. So we have peptides that work as biostimulants for crops like soy, cotton, that we've now taken to market and we've commercialized, right? So so where are we? It's been 10 years of building the tech. But what we've also shown in terms of String is, I think we've clearly established a state-of-the-art technology platform using genetically modified and non-GMO strains to convert methane gas into very, very interesting products. Today, I would say String is a world leader in terms of gas fermentation technology. We have about 60 plus patents that cover various aspects of this core process. We've also built a pilot scale facility about 5 years back. This is a 1,000-liter fermentation capacity facility that we built. And using this facility, we brought our products to market. We provided to customers. We allowed them to test. given that we're making protein from greenhouse gases like methane and carbon dioxide, we gave it to potential customers to taste and see, hey, how is our product going to do in the marketplace? And then about 2 years back, we set up a large-scale commercial facility, which we today build, own, operate. And using this facility, we have now launched our products commercially in the market. Our products are profitable and using a biology-based platform. So why am I sharing all of this with you today? Well, I thought, I'll change and focus on some of the questions and points that really bother me with a community of like-minded people. Because I've been in the synbio space for about a couple of decades now. And I have to say, even though we've done a lot of work at String, the journey at String has not been easy. In fact, it's been very, very difficult to go from having a very interesting technology to building, scaling it up to a pilot scale, and then taking it further to commercial scale and taking it all the way to market. So rather than talk about the science, which is very, very cool, and I'd love to talk about it endlessly, I wanted to perhaps pose a couple of questions to the audience here that we can perhaps debate over dinner and maybe drinks later in the evening. The first question is that in the last couple of decades, one of the things that I've observed is while we as a community are building some very, very fantastic technologies, we tend to focus too much on technology and perhaps not enough focus on how we will build businesses and bring value to the marketplace using this technology. So one of the first questions that I want us to perhaps ponder about is how do we graduate as a community from building technologies to delivering value-added businesses? Why is it important? Because I think, in our community, we have some very interesting technology that we start with, but by the time it goes to the marketplace, we're not able to deliver the same kind of value that we promised. And why is that? I think that's because there's a lot of very, very critical market-side questions that are being solved one at a time. Right? That's being solved by us in India, some other company in China, some other company in Singapore, a couple of startups in US. We're all trying to solve these market-side questions individually, and we really need to be solving it perhaps together. I will give you an example, right? Let's talk about when is product market fit established for a bio-based product? Now, suppose you're making a bio-based product using a genetically modified organism. At what point do I tell my investor that, hey, this product market fit is established for this product? Is it when I run a 5-liter fermenter, I make that product, and I give it to a customer and the customer says that this product is fantastic. At that point, is PMF established? Or is it when I put it in a 1,000-liter fermenter at pilot scale, and now I've given it to maybe 50 customers, and I have, 10 gram per liter titers. If I get it to 30, I can see economic viability. Is that when I have PMF? Or is it when I actually scale it commercially? I'm running a 10,000, 20,000-liter fermenter. I'm making my product, whether it's a small molecule, it's a dye, it's a biofertilizer. I'm making this at 20 gram per liter, and now it's commercially viable. At this point, is it now when I can say, hey, we have product market fit? Right? And these are all questions that each of us are solving separately, but we really need to be thinking about this more as a community. So one of the first questions that I would love to perhaps have a discussion about is, when does a synthetic biology company move from being technology-focused to saying, hey, I'm able to deliver value in the marketplace. The second point I wanted to highlight is that while within our ecosystems, right, we all are fascinated by the science, fascinated by the tools we are building, fascinated by the promoters, the sensors, the capital markets, that is the investors who have to come in and help us scale and bring these technologies to market, and the consumers who then have to buy these technologies, they are very, very unfamiliar in how we can add value to their lives. For example, Ron here talked about how microbial production using, for food using CO2 is very valuable for humankind. We do something very similar. We do food production using methane, and it's very exciting. But 95% of the people that I talk to outside of this ecosystem do not understand the value of that. So the second question I want us to think about is, could we have a standard framework that helps others who are not within this ecosystem evaluate and understand the value proposition and the value that we're able to generate, right? So let's say you have phases. You have an ideation phase when you have your genetically modified E. coli, your Saccharomyces, and it's considered a very high-risk phase. At that point of time, you can work in a lab, you can work in an incubator, you can make that strain, you can run your small-scale vessels and demonstrate that, hey, this technology can work. Right? If you think about it in terms of ChatGPT, that's your very, very first algorithm that somebody is writing. But maybe from there, there's a pilot stage, right? And then there's a commercial stage. And then there's an actual growth phase of the business where you've taken your dye or your small molecule or your drug or your food product to market, demonstrated that somebody will pay money for this, demonstrated that you can sell it profitably, and that you can grow the business around it. Right? At this point, you have investors who then can come in and say, okay, what is the actual value of this business? What is the annual recurring revenue for this business? Is it profitable? Is it scalable? And these are languages that others can talk about. So maybe as an ecosystem, we need to simplify it for others around us and say, hey, here's work at an ideation phase. This is the risk and this is the value. Here's a work at perhaps a pilot or commercial phase. These are the risks. And if this risk gets mitigated, this is the value. And here's what a growth phase business can look like. Because this is something I've seen personally. This is the fourth startup company that I've been part of. And it's very, very, very difficult to communicate the risk around what we do to folks outside. And perhaps the question is they don't want to understand. They just want to know what is the risk and at what point will you deliver value. And maybe we need to make it easy for them by perhaps coming up with standard frameworks, right? The last is that, again, there is a lot of interesting technology in the early stage of a SynBio journey. But over the last 2 decades that I've been part of this community, there's only a handful of success stories that have been able to go all the way. In fact, when my investors ask me to point to other companies, SynBio companies that have been successful, one of the companies I point to is almost 25 years old, right? We don't have too many companies that have really been able to show profitable value in the market in the last couple of decades. I know I'm the messenger of bad news here, but I do want to, that's the reason I want to provide context, and I want to share it as a safe space where we can have open discussions around some of these really critical points. There's been a lot of companies that are running this race and are close to winning, and there's been a lot more failed attempts. So one of the things that I really want to pose a question for the audience here is, given all the fantastic technologies we have, could we as a community put frameworks in place that allows for these companies, for these ideas to really scale in a much more robust manner, such that within the next 10 years, that is by 2035, we can actually have at least 10 synthetic biology-based companies, each of which have been able to demonstrate at least $1 billion in revenue and have demonstrated profitability at scale. Right? So one of the challenges that I would like to pose for ourselves is, could we have 10 by 10 by 2035? That is 10 SynBio companies. I don't know if you guys saw, NVIDIA has a value of almost 5 trillion, right? Where are the equivalents in the synthetic biology space? So could we challenge ourselves to have 10 synthetic biology-based companies, each of with at least $1 billion in profitable revenue by 2035? And given what I've seen, I think being able to have predictable systems that I think somebody earlier talked about, having rapid scalability and having sustainable businesses, I think is very, very critical for us to be able to get there. So just to wrap up, at String, the journey for us started with an ideation phase. So we had a very robust technology platform today. We then took it to scale. So we built scalable infrastructure. Unlike a few of our competitors who perhaps worked with CMOs, we decided to build our own plants. So today we have a large-scale plant that is online. And now we are working with strategic partners globally to bring these solutions to market, right? And the journey, I have to say, unfortunately, has not gotten easier. Even today, with demonstrated manufacturing capacity and profitability, we do continue to face headwinds in terms of growing the business, right? So I'll end on that note, and looking actually forward to perhaps having discussions later in the evening with some of the folks in the audience and seeing how we could brainstorm together to help more companies scale and maybe do it in a faster and more sustainable manner.
Thank you, Ezhil. Thank you so much. So there's a microphone here, so raise your hand if you have questions to anyone or to all the panel. David has a question. I have a question for all of you. So one of the key aspects in iGEM, and I think part of is exemplified in your work, is sharing, right? There is a lot of complementary work here, right? If I could have magically, put the diversification system of David, right, into Ron's train, use all the computational informatics, right, in order to assess which genes should go there and which ones we should discard, right? Things could accelerate much faster. So maybe a few words about, and I think you're excelling in sort of communication and sharing in your labs, and you have several tools. So maybe shout out a few examples from what you do here that sort of makes this happen and do this combination. And for Ezhil, I'll turn the question to the other side, right? So you imagine an amazing world in which we'll have 10 amazing companies, each one 10 billion, right? And what we see in synthetic biology is when synthetic biology really started rising, right, is it was not 5 big labs that expanded, right, but there was explosion of talent, right, and hundreds of smaller labs that managed to work together and do this. So from a model of sort of 5 big companies of 10 billion to hundreds distributed in an equitable way over the world companies, where do you see this going, and what's the best advantage? So quickly, David, Aude, and Ron, and then Ezhil, please.
So, yeah, I mean, clearly we're in a world where all our projects benefit from sharing from multiple disciplines, and I think we've seen initiatives like iGEM having a huge impact on that. I think in the CRISPR field in particular, I think it was both ways, right? The CRISPR field helped launch iGEM because so many people in the CRISPR community were sharing their tools there. And at the same time, iGEM helped so much spread all these tools and make them available to our labs. And so I think this kind of initiative are super important. No, I think there is a lot of work to do to also make all the bioinfo tools more available. And I'm sure Aude will be very happy to comment on that.
Yeah, I think for me, a lot of people are scared to share, thinking that they will lose their edge, and I think it's the contrary. In all of our fields, I think when we start thinking like, how can I empower the community and convince them to work on the topics I think are important, it feels like hundreds of people are actually tackling the same topics. So for me, there is really a powerful aspect towards building communities and to putting in as many hands as possible the tools. And it's true in my lab, we do a lot of bioinformatics prediction, and we constantly have in mind that these predictions are just generating so much work for experimentals that we could never even tackle them by ourselves. But beyond that, I think that for me, the key thing is not only to give predictions, but give the tools so that people can make their own exploration, because they will take it to a way and places that we would never have thought of, and that's when great discoveries are made. So yeah, we try our best. So you have the classic stuff like wikis, of course, sharing as you mean, Git. I mean, you have tons of solutions that exist right now, and with now all chatbots and things like that, talking in a natural language to a lot of computers has become much easier. So I think we're living in a fascinating moment where a lot of these become easier, and a lot of people will have even more kind of superpowers thanks to all these tools that are being shared.
So it reminds me, so clearly here, even just in this panel, I'm very curious about, getting stronger tools from what was discussed here about the ability to, evolve better, build better, and also to learn from your real-world experience, the fight, to really understand, what needs to be happen to make things economical and scale. So we'll continue that discussion as you suggested, but it just makes me reflect on, so because this was a long-term project, when we were at the point of we celebrated 10 years of this effort, and then that was roughly when we were already successful to achieve, this significant milestone. And then we said, okay, let's look back and reflect, what do we understand now, and what enabled those steps to happen. And it was interesting. I had all sorts of people from around, many years, and it turned out that the jumps were in places where actually people from outside the lab came to visit the lab, work in the lab at the Weizmann Institute for some of them were coming just for a few days, some of them were coming for months, but this is the thing that, got us out of things we were stuck in. And actually, I didn't understand the importance of that until we did this, long-term reflection. So that was very clear that this was essential, and otherwise maybe, we'll still be without any achievement.
Thank you, Ron. Ezhil, sorry to put you on the spot, but I'm sure you've given it a lot of thought.
So I want to say that if we enable at least 10 such successful companies like that, it is but inevitable that will trigger 100 such smaller companies. It's typically what I've seen happen is once you have a very successful company, the talent and the solutions that it created, that kind of creates folks who come out and then start to build around it, and then they build some more. So it's never a sharp, we'll never have a future where it's 0, 0, and then suddenly 10. We'll have a tail. So my vision is if we get to that 10, you will have a longer tail of smaller distributed companies around the world. It's only but natural because that's how nature works.
If I can add a few words on that, I think much more is going to happen. So I'm, way beyond the 10-10 challenge. Because if you look at what's the prediction on what is needed to happen to get us anywhere near net zero or something like that, the estimate, let's say from McKinsey, but there's different estimates, is in the $300 trillion between now and 2050 in terms of what's going to happen here. So if you take that, that's about $10 trillion per year. That means the 10 doesn't have to be the billion, it has to be the trillion, not only in the synthetic biology, but in terms of the scale of the challenge. And the scale of the challenge, I wouldn't be surprised if like synthetic biology would be a part of that. And I agree that so far we're not seeing it accelerating at the rate required for that. But this does not mean that the need is not there. It just means that humanity is not moving, civilization, society is not moving anywhere fast as needed, but nature doesn't care. You know, those challenges, you know, nature doesn't look at, you know, what's the politics, etc. It knows what's going to happen in terms of climate, in terms of the ecological crisis, in terms of the needs of water supply, etc. And therefore, something is going to happen, and we better adapt for that, and maybe the potential is much, much bigger.
Thank you, Ron. Question for you? Go ahead.
I actually had a question for Ron. I was wondering if you had any thoughts on potential impact of fission and eventually fusion energy on the economics of what you were showing.
Yes, so, you know, the potential either of fission, if, you know, humanity starts to get back to it, you know, very much, or fusion, which would be wonderful in many ways. It's, you know, it's a very promising thing. I wish things have moved faster. Maybe that would be the transformative solution, but I think in any way, we should not build just on that, because really this is something that's very hard to predict. And we should also remember this would be very helpful, but this is the energy aspect. This would not, you know, we have several crises and several needs in parallel. This would help a lot, but we have other big challenges. This does not supply food by itself, and therefore we need to work in, you know, we should not get into the mentality of just hoping for like a miracle-like technological solution of that sort. We have to work on several technologies and also on, you know, policy making, behavioral changes, etc.
Time for one question from the audience. Do we have one? If not, thank you so much for everyone. Thanks for the panel, and we'll move for the final panel. Thank you so much.